Thank you so much, Megan, and thanks everybody for dialing in. We really are thrilled to have you here, and we hope that you can get the most use out of our webinars as an option, for you to get technical info without actually having to leave your home and in these times, as Megan said. So let's dive right in. So today we'll talk about the AmpliSeq for Illumina solution. So I will walk you through the available products. We will talk about kit options as well as the workflow step by step. We will go into our Design Studio software and talk about how to use this software for custom panels. We will go over the available analysis options, and I will leave you at the end with some additional resources. So with AmpliSeq for Illumina, we really wanted to give you an efficient, simple workflow with minimal hands-on time for this kit. It's only 1.5 hours, only 10 steps. Overall time, about 5 to 7 hours for the library prep, so you could do this on day 1 of your experiment. Day 2 could be reserved for sequencing. Depending on the platform, this could be roughly between 17 to 32 hours. We, of course, you might be familiar with our load and go reagents and our push button sequencing solution. And then day 3 could be reserved for your analysis. This time for the analysis will vary depending on your panel. And we have local and cloud options. And all of our analyses on the cloud or the local options that we give you are as automated as possible to really make it usable for you even without an extensive bioinformatics background. So let's start by talking about our available products. We have ready-to-use off-the-shelf panels in the DNA, the RNA, as well as the DNA and RNA category. So these are, as the name says, ready-to-go panels that you can order and use without having to think about your own custom content. We have those predominantly in the cancer area, but also we have an exome panel, in the immune field, some panels, and several solutions here. So these are great if you perhaps don't have an exact idea of your content, but you know which area you're working in. Another option is to use our gene panel and array finder tool. If let's say you have a gene of interest or a few genes of interest that you definitely want to have in a panel, but you'd like to use a off-the-shelf solution, you can use this tool to check if any of our kits contain your gene of interest. Now, if you have a really good idea of exactly what content you would like to have in your amplicon panel, you can do a custom design. Here we have the option to do custom DNA panels, so you would go and use our free Design Studio software to choose your targets. We have a variety of genomes available, and we have options for full custom or so-called on-demand and on-demand spike-in options. And I will talk about what those solutions are in a little bit more detail in the coming slides. We also have custom RNA panels as well as custom gene fusion analysis. Again, also, you would start in Design Studio, and you can choose from pre-designed amplicons. These are actually going to be from our human transcriptome expression panel. So you would create your subset of exactly only the genes that you would like to have included. And we have more than 1,000 well-annotated fusion genes available for you to choose and to pack into your custom RNA panel. You can enter genes by the name, the transcript ID, or you can use lists to upload to create your panel. And then we have our community DNA and RNA panels. These are panels with custom content that were designed in consultation with experts in the field. And we have options for bacteria, viruses, as well as humans. So let's talk about the workflow. As the name says, AmpliSeq is an amplicon assay. You would start with either DNA or cDNA generated from RNA through reverse transcription. And then the defining step here really is a big multiplex PCR, where you are using primer pairs to amplify your regions of interest. This is then followed by a digest of your amplicon. So that's where we're removing the primer sequences again. Of course, these primer sequences themselves were not generated by filling in between the primers. So we don't want to use these for variant calling, of course, since we would only be calling variants really in our primers, which wouldn't make any sense. So we are removing these in a digest. And then we are ligating adapters to our insert. And these adapters will contain all of the sequences crucial for flow cell binding and sequencing on the Illumina platform. And this is followed by a final PCR amplification to create our sequencing ready fragment. Let's talk about the reagents that you would need. So there's 3 classes of reagents. First, you would need a library plus kit. That kit contains essentially all of the library prep reagents. We have a 24 reaction kit, a 96 reaction kit, and the 384 reaction kit. This would then be paired with a panel. The panel includes the primer pools that are necessary to amplify your regions of interest. And here we could have a so-called one pool design, or also called a hotspot design, or designs with up to 12 pool designs. Although those are rare, the 12 pools really only our whole exome solution. Most of them will have 2 pools or maybe 3 or 4. And we'll go into that in more detail in a couple of slides as well. And then the third thing that you'll need is the index kit. So here we have a couple of options to choose from. We have the combinatorial dual or CD indexes, set A, B, C, or D, with 96 indexes for 96 samples. And then you have the option to purchase set A through D with a single catalog number altogether. That's our set A through D with 384 indexes for 384 samples. Then we have the CD indexes as a large volume solutions. Those will be set A. So that is a great option if you're using automation in your lab. And then we also have the UD or unique dual indexes with 24 indexes for 24 samples. We have support pages that talk in detail about the shipping and storage conditions. What I wanted to mention here is one thing to note is that some of our reagents ship at a different temperature than how they are stored. So that's something to pay attention to. For instance, the oligos and the indexes ship at room temperature and is perfectly fine. But then once they arrive, we want to store them at minus 25 degrees, or so basically in the freezer, minus 20 degree freezer. One more thing to note is that the number of reactions doesn't always equal the number of final libraries. This is the case for 1 or 2 pool designs where a 24 reaction kit will give you 24 libraries. However, if you have some of our larger designs that have 3, 4, or more pools, you will get fewer libraries than the reactions in the kit. So this is something that's important for project planning. So for instance, with a 3-pool design, with a 24 reaction kit, you'll be able to prep 16 libraries. And the reason why this is important is that with such a multiplex PCR, we have to keep our PCR primers separate into separate reactions so that they don't interfere with one another during that multiplex reaction. So we are, at that point, we are using, the workflow will be outlined in the protocol in greater detail. And another note is that the DNA and RNA samples are prepped separately, so each preparation would be considered a unique library. And a single sample can give rise to 2 libraries, a DNA and an RNA library. So let's do a deeper dive into the workflow and go step by step. So we would start by getting an accurate quantification of our DNA or cDNA and then diluting that DNA. This is followed by the amplification of the targets, the partial digest, the ligation of the index adapters. Then we have cleanup, followed by another amplification of the library, a second cleanup, and then our QC and normalization steps to get us ready for sequencing. So the first thing you would do is prepare your input. So your DNA or RNA would be, your RNA would be converted to cDNA. And here we would use the AmpliSeq cDNA synthesis kit. Our typical input is 10 nanograms per pool. So keep in mind that for a 2-pool design, this means that we will need 20 nanograms. But there is certainly a range that's acceptable. So we can be anywhere between 1 to 100 nanograms of input here. There's actually no limit on the quality of the input. So FFP DNA is accepted, and we actually know the kit does relatively well with degraded input. So you would set up your PCR reactions by combining the DNA with the hi-fi mix and the water. And all of these steps are detailed in the user guide, so you can follow the instructions there also step by step. You would make an aliquot of DNA, a master mix, and then add that with the primer pools 1 and 2 for a 2-pool design, or as appropriate for your experiment. And then we would set up the multiplex PCR. The PCR cycles and extension times vary depending on pool size, number of primer pairs, and we have that information listed in the reference guide, as well as the quality of the DNA and the input amount that was chosen. And again, in this reaction, this is really the critical reaction here, where we are generating our amplicons, defining our regions of interest. This is then followed by the enzymatic digestion of the primers. Here is also where we can combine our pools. Now that the amplicons are generated, there's no need to keep them separate anymore because they're going to be sequenced together. This separation is really only necessary during the PCR, where we are otherwise at risk of having the primers interact with one another and potentially forming undesirable amplicons. And then we are preparing for ligation with that partial digestion of the amplicon ends and the phosphorylation of the 5' ends. This creates blunt-ended phosphorylated fragments for our adapter ligation. So now we would ligate our indexed adapters. This requires the reagents switch solution, ligase, and our indexed adapters, of course. As mentioned previously, we have a couple of specs to choose from. We have our 96-plex combinatorial dual indexes. So these are all unique, all combinatorial. So some indexes are repeated between the I5 and the I7. If you would like to go with unique dual indexes, so none of the indexes being repeated between samples, we also have the 24 unique dual indexes available. Note that with that plate, only the first 3 columns of the plate will be filled. So the rest of the plate is empty, and that's designed. So because we only have 24 indexes here. And again, with the CD or combinatorial dual indexes, you still have unique index combinations, but some indexes will be repeated between samples. Another resource that I have a link here for is our index adapters pooling guide. So that's something that you can consult for project planning as well. This is a 3-step thermocycler program for this PCR, for this reaction, where we are ligating our adapters. This is then followed by a 1x bead cleanup. Here we're using user-supplied Ampere XP beads, and we're using a 1-to-1 ratio to remove any fragments of less than 200 base pairs, but just to clean up the reaction. And there's a neat resource here shown on the right side, how the ratio of beads to reaction volume changes what is retained on the beads and what is washed away. And with that 1-to-1 ratio, we're basically only removing anything, such as adapters that we don't want in our reaction. Here we want to use a Dynamag 96 side magnet for the best results. And after the ethanol washes, there's a step to air dry the the pellet and then proceed immediately to the next steps. I know this is always tricky to do these ethanol washes. It's always hard to know how long do I dry to really get the perfect amount. And what I wanted to mention here, it's actually better to slightly over dry than to carry the ethanol over. So of course we have instructions in the reference guide, but if you're ever in doubt, this is good to keep in mind that it's okay to slightly over dry. We would then amplify our final library. Something to keep in mind here that can be confusing because we don't usually have that in our workflows is that the beads remain in the reaction. So that's completely fine. We are using a short 7-cycle PCR just to generate enough yield for sequencing as it is shown here. And this gives us our sequencing ready fragments. So then we have our final double-sided bead cleanup. And let's talk about that in a bit more detail. So we're starting with a 0.8x ratio to remove the large DNA, and that's followed by a 1.2x ratio to remove any small fragments to get our final library in the desired size range. So here it can get confusing, so it's important to remember what to discard and what to keep. We're first adding beads to the sample, vortexing and placing on the magnet. And keep in mind that this step actually adds beads to the beads that are already still in the reaction. So that is correct. Here we want to keep the supernatant and then transfer that to a new well. Then you're adding beads once again, vortexing, placing on the magnet. Here we're removing the supernatant and keeping the pellet. Washing twice as outlined in the protocol, and then we can elute in low TE buffer. Okay, then we have our final library, and of course we want to assess the quality. So here we have a couple of options. You can use a fragment analyzer or a bioanalyzer. Typical libraries show a size distribution from 200 to 275, but of course that will, especially for custom libraries, it will depend on the choice that you made in the beginning of your design for your desired insert size. Keep in mind that when you're looking at your library on a trace, our adapters are adding about 135 base pairs to the size of our actual insert. Then we want to quantify our library. Here are lots of options, and all of those are acceptable. You can quantify with a fragment analyzer, bioanalyzer. In tech support, we have best results really with either fluorometric methods such as Qubit or AccuClear, PicoGreen, or Kappa qPCR is also a great quantification method, but all of these are acceptable. Then, in order to prepare for sequencing, we have our typical dilute and normalization steps. So we want to go from 2 nanomolar down to 20 picomolar. We have a pooling calculator that can help you help you with the math to get down to 2 nanomolar. And then we really have pretty detailed instructions in your instrument's denature and dilute libraries guide on how to do this denaturation, the dilution, and even recommendations for final loading concentrations for your sequencer, as well as some recommendations for DNA to RNA ratios to make sure that you get the appropriate amount of reads, because our RNA libraries tend to use fewer reads than the DNA solutions. For sequencing, the consideration is, of course, the run length. As for typical amplicon sequencing considerations, we want paired end reads that should overlap in the middle. For most ready-to-use panels, we want to use 2 by 150 cycles, and this information can be found in the library prep and array kit selector for our off-the-shelf products, and there's a link here on the slide. For the custom panels, it will depend a little bit, again, on the amplicon length that you chose in the beginning when you designed your custom panel. For 140 and 175 base pair inserts, we want to use 2 by 150. 275 should use 2 by 250. 375 would use 2 by 300, and that ensures, of course, that we're not missing anything, that we're covering even our longest amplicons in the design. And 2 by 300, of course, is only an option on our AmpliSeq instrument. We have automation solutions for our AmpliSeq kits. So we have methods that were developed by our leading vendors, and we have an automation page that have links to our vendor pages where you can find more information about scripts. And there's a link here on the slide. Let's talk a little bit about additional either required or accessory kits. There's the AmpliSeq cDNA synthesis kit that, of course, is needed if you have an RNA panel that converts the RNA to cDNA. Then we have the AmpliSeq for Illumina Direct FFPE DNA kit. That's optional, but a great thing to use if you are preparing DNA from FFPE tissues. It does not require deparaffинization or DNA purification. And this has actually been specifically developed for our AmpliSeq for Illumina protocol, so we unfortunately can't use it for any other Illumina library prep kits where you wanted to use FFPE tissue. We also have the AmpliSeq for Illumina sample ID panel, which is essentially a human SNP genotyping panel that creates a unique signature for each sample that is pre-bundled in some of our off-the-shelf kits, such as the BRCA kit, the comprehensive V3, and the myeloid kit. But it can also be used with our other kits, and it is spiked in at the amplify target step. So that creates a signature for each sample, as mentioned here, and can help detect any potential contamination. We also have the ERCC RNA spike-in accessory kit. That is, as you may know, a set of external RNA controls developed by the ERCC and hosted by NIST. So these are standards that are sometimes used in RNA expression analysis, a set of unlabeled poly A-tailed transcripts that can be added to the RNA right at the beginning after the extraction or isolation. And the purpose is to measure sensitivity or dynamic range of an experiment and to help quantitate differential gene expression. But this is also an optional panel. And then we have the AmpliSeq Library Equalizer Kit for Illumina. Excuse me. That is a kit for final library normalization that can be used if you are not wanting to do manual normalization, also optional. It's a 96 reaction kit. So let's talk about that in a bit more detail about how this works. So it will be incorporated in the workflow. So in your reference guide, there's a whole section that talks about the equalizer workflow, because there are some modifications in the workflow that you'd want to make even during the workflow if you are choosing to use the equalizer to normalize your libraries at the end. So this protocol requires a few more PCR cycles at the Amplify library step just to make sure that we have enough material to do the equalizer protocol. Keep in mind that the resulting libraries are double-stranded DNA. And I'm pointing this out because you may be familiar with some other bead-based normalizations that we have for other Illumina libraries, where the resulting libraries are actually single-stranded. But that's not the case here with the equalizer. These libraries will still be double-stranded. But no need to worry, you will be guided through how to use the equalizer, how to denature your libraries, and how to load with our reference guide, as well as the denature and dilute library guides. They have a specific section for AmpliSeq, either manual normalization or equalizer normalization. Great, so now that we talked about the workflow, let's shift gears a bit and talk about how to make a custom panel using Design Studio. It's one of my favorite products that we have. So I wanted to mention as well that we actually have a dedicated webinar for Design Studio coming up soon. So if you would like to go into even more detail, that's a great webinar to sign up for. So the design process really was made to be very intuitive, a wizard that you would just follow and click through. You would start by choosing your assay type. This is either DNA or RNA. Then you choose your technology. We have AmpliSeq for Illumina gene, hotspot, or on-demand. And we also have the enrichment solution, which is a different library prep kit. We have different species available, human, mouse, so 2 reference genomes for human, as well as some extended species. So let's talk about this, hotspot versus gene design. What does that mean? For a hotspot design is generally for small genomic regions such as SNPs or small indels. And what that means really is that a single primer pool will be able to fully cover your region of interest. So that means that we can put all of those primer pairs into a single tube because we really don't have any interference between those primers, generally in a hotspot panel. Whereas in a gene panel, as you can see here on the right, we might have a target that is so large that you may need, let's say, 3 primer pairs to fully cover that region. In that case, neighboring primers would create undesirable off-target amplicons. For instance, one of the red primers shown here could create an amplicon with one of the blue primers, even either a really small or a much larger amplicon that we don't want. So we need to separate these out into 2 pools for the PCR reaction. And also, with our custom panel, we can have between 12 to 3,072 amplicons per pool. So on the next step with Design Studio, it will ask you for your sample type. So this could be either regular, so that means high quality DNA, or RNA, FFPE, or a cfDNA. And this will really limit your maximum amplicon length. That's the only difference here, because the more degraded your DNA is, the smaller your maximum amplicon length should be in order to give you the best success with your panel. So with cfDNA, which we know is very short, we're actually only allowing a maximum amplicon length of 140 base pairs. Whereas if you choose regular, you have all 4 options available for the maximum amplicon length. Then this takes you to your input design targets page. And we have a couple of options here. You can choose to add your genes by gene name. We have options here to choose either coding sequence or exon only. The exon only includes the coding sequence as well as the 5' and 3' UTRs. We have the option to add coordinates. And one important thing to note is you want to always use CHR and then your chromosome number at the chromosome field here. And also make sure that you are really basing the coordinates that you're inputting here on the reference genome build that you chose at the very beginning. You also have the option to upload a file, so especially for very large designs, no need to go one by one by one. You can download a template, enter all of your input, and then upload that file to basically get all of your targets added all at once. And then we have this Copy by Amplicon and the Community and Fixed Panels tabs. So these are pretty new. So that's a great feature. Let's talk about that in a little bit more detail. So this feature allows you to copy existing amplicons from a previous or from a community design. It's what we call whitelisting of amplicons. So basically this is what this looks like, and this is the information that we need from previous amplicons in order to whitelist them and to get them into your design. So we want to add, and the most important things are the amplicon ID and the solution ID. And here's what the screen looks like for if you're choosing a community or fixed panel, where you can copy either all amplicons or some amplicons over to your custom design. Okay, so once you've entered all of the information, you submitted your design, you will get an email when the design process has completed. So the algorithm does its thing to generate all of those primer pairs, cross-check them to make sure we get the best in silico coverage. And then you will be linked to a screen that looks like this, where you can review your design. You get the overall calculated in silico coverage, as well as things like the total target size, the number of pools, the amplicons per pool, and such. Also the minimum, maximum, average, amplicon length. Another new thing that is an option here is this button for See More Solutions. So the algorithm will choose what it thinks is the best design to show you here. But there's also alternate designs that you can review if you like. For instance, if there's something about this design that doesn't meet your expectations, you can also go and look at other solutions and how they differ. However, rest assured that it will automatically choose the best design for most users right here on the Review Design screen. You can also modify your design, and that allows you to give the design a new name, essentially create a copy, and for instance, add or delete any targets to that design, basically to a copy of that design. So your original design will remain untouched in your account. So let's talk about the on-demand panel. So these are more than 5,000 predefined genes. And the cool thing about this is that these are wet lab tested and there's performance data with them. So we have this for a coding sequence, not for intrands or UTRs. And generally we have smaller reaction sizes. But with that solution, in addition to getting your amplicon, you also get some performance data along with it. Now, if you would like to use the on-demand option, but maybe some of your genes aren't available on demand, that's where the spike in panel comes into play. So you can basically combine on-demand with brand new designs and still get the best of both worlds, so to speak. So get some amplicons on demand, and then for the rest, you would get your custom design. So we often get that question about how many samples can you sequence. So we have a couple of resources to help with that. Of course, it's never an easy answer for a completely new custom design. We have a coverage calculator on the Illumina support page that has most of our off-the-shelf solutions preloaded that can give you some guidance here for DNA as well as RNA. And then we also have a calculator under My Illumina when you go to More, My Tools, and Custom Products to get some more help and in regards to what to order and how many samples can be sequenced. So this is what that calculator under My Illumina looks like. For custom panels, you would enter your number of amplicons, your number of samples, and your project type. And then click on Get Pricing and Package Details. And then you get some and this is how it looks like for the community panel. And then you get some suggestions on how many samples to and how many reagent kits to purchase. And you, of course, also want to put in your mean coverage steps. That's something to decide. That will dictate, of course, how many samples can be pooled on any given sequencer. And here's the output, again, giving you some suggestions on what Library Plus kits to obtain for the experiment, as well as index kits and accessory products. Okay, so now that we talked about Design Studio, and again, in the upcoming webinar, we'll go in a little bit more depth in how to look at designs, how to review regions as well as gaps, and how to also check if the design meets your expectations. But we'll switch gears here a little bit and talk about some analysis options for your panel. So we have applications available both in our base-based sequence hub, so that's the cloud, as well as local run manager. We have the DNA amplicon app and the RNA amplicon app, or the module, as it's called on local run manager, to analyze your DNA or RNA AmpliSeq for Illumina panels. On BaseSpace, we also have the CNV analysis app, as well as the Pindle app that is great for indel detection. So let's briefly talk about the DNA amplicon analysis. So it uses FASTQ files as input. These are produced after demultiplexing from our sequencer. And it analyzes your DNA that is enriched for specific targets. It does an alignment of the amplicon reads against a reference that's specified in the so-called manifest file that's provided to you in Design Studio. And then we're calling variants for all of our target content. And we get reports for coverage statistics for each amplicon, as well as a summary of all the overall metrics. And this is available, again, as I mentioned before, in BaseSpace or locally on LRM. And this is more of a resource slide for you that talks about or lists the output files per sample that you get. Alignment data is captured in BAM files. Variant data is captured in VCF files. And then we also have genotype data as well as statistics reporting that lists all of the results coming out of the analysis. Now the RNA amplicon analysis. Again, we're using the cDNA that is enriched through our AmpliSeq reaction for specific targets, aligning the reads against a reference, performing differential gene expression as well as fusion calling. And then there's reports of read counts mapped to each gene for each sample, and we get a nice summary report available on BaseSpace and on Local Run Manager. Here's a list of the output files per sample for the RNA amplicon app. We have statistical reports as well as gene expression and fusion data. That's really what we're interested in with the RNA solution. And the different file names, types, and description is listed here on that slide. The amplicon analysis report also contains data as shown on the slide here, and that can be sorted to get more expression, differential expression results for different genes. And you can also, this is a searchable field also, where you can go and filter for your particular gene to do more of a deep dive on the expression results for different transcripts. So let's talk about public data and manifest. I've already mentioned manifests earlier. So first of all, BaseSpace Sequence Hub has a lot of public data sets for our ready-to-use panels. And these are really a great resource because you can get a really good idea of how AmpliSeq for Illumina performs, what runs look like, what the analysis looks like. You can browse through all of the different analysis files and decide whether this is intuitive. If you have any questions, you can always call us a tech support and see if this is a good solution for your experimental approach. And it's completely free to import into your design, into your BaseSpace account. The manifests contain all of the information about the targets for our AmpliSeq panels. For our ready-to-use panels, they are available on the individual support page. For some panels, you may need to call or email tech support. And for your custom panels, they are downloadable in Design Studio after you've completed your design. Here are some additional resources. So some links to our library prep and array kit selector. We have great frequently asked questions on the Illumina website, links to support pages, as well as some more resources for Design Studio. And then my last couple of slides, again, are intended to be resources for you, basically a summary of the different ready-to-use panels here for DNA with all of the most important information, such as information about the input, how it was chosen, the number of pools, the number of amplicons. Here for the RNA panels and both DNA and RNA that hopefully will help you choose the panel that's right for you. And of course, at Tech Support, we're always here for any questions that you might have. And that brings me to my last slide. And thank you so much for bearing with us and for your attention. And we're ready to take any questions.